Hello, I'm Dave Tabb, and today I'd like to talk with you about web resources for plant biotechnology. This lecture was originally created for an Open MS roadshow we did in South Africa to cover 4 different universities down there, and so I was really trying to find the best set of web resources that people who are new to the field, maybe coming from human biotechnology, or from other areas, to learn the basics of what they need to know to find genomic resources and proteomic resources as well, and metabolomic resources across the space of plant biotechnology. So it's intended as an introduction, and maybe even if you have some experience in the field, this might tell you about some resources you didn't know about. So I hope that you'll find it useful in getting you started. We're going to start with some resources spanning a very wide variety of plant species, those being phytosome, ensemble plants, and plant garden, a relative newcomer to the field. And I hope that these are useful in letting you know about web resources that may not be obvious to everyone in the field. But it's also important, I think, to talk about model organisms. And in that respect, I wanted to highlight 2 specific, species-specific web resources, TAIR and RAP. So we'll come to those in a moment when we talk about Arabidopsis and rice genomes. From there, I'd like to talk about some that are specialized to particular crops that don't get a lot of attention. And for that, I'd like to talk about the African Orphan Crops Consortium. They've done some really good work in species that you may not know about. And then finally, I want to talk about how you can find existing experiments that have already been produced in metabolomics and proteomics for these species. And for that, we'll take a quick look at Omics.DI, a search engine. Okay, so let's talk then about phytosome. Phytosome has been a big player in this space for quite a while, and it's worth talking about why it's had such enduring value for the field of plant biotechnology. So for this, I would note that phytosome has 188 different species that have been studied. By the way, if you find that the stuff appearing on the wall behind me is not clear enough, I strongly urge you to download the PDF of these slides, which should be available in a text description below this on YouTube. So 439 genomes have been sequenced now by phytosome, and they span 188 different species. That's a great deal of power. And even better, they've already pre-computed orthology, which is to say that if you have a gene in a relatively less studied species, you may be able to find that it is similar to the same gene in other species. The same gene has separated to two different species, and then in those species it's independently drifted, and yet we can still recognize it by sequence homology and thus figure out a function for this gene in a new genome because of its similarity to one in a well-analyzed genome. Okay, so computed orthology is available for these. Maybe the gene you're interested in doesn't have a homolog, but maybe it does. So that's what this pre-computation of orthologs is so good for. Now, the next thing that you might want to know is, can I attribute a function to this gene, for example, within gene ontology, or by its content of particular domains that are known to be associated with particular functionality. So COG, KEG, enzyme, pathway, and interpro have already been run on all of the sequences appearing in phytosome genomes. So that's a really great bit of power. If you had to run interpro yourself on a genome, well, then you need to download the several gigs of interpro scan software to run it locally. So it's really powerful that they have all these resources already available for you. More recently, there's been this really cool project called the JGI Plant Gene Atlas. And what they've done here is to add expression data on top of the genome data that we already had. So that's really powerful to have some indication of which genes get most expressed or have the least measured expression. That could be a hint as to which ones are functionally active genes and genes that are not active. So having that information is very valuable. Now, I've spent a lot of time here talking about phytosome as this canonical resource for plant biotech, but there's this little challenge that plant genomes, you would think, would be part of, for example, the Agricultural Research Service of the FDA. But in fact, that's not who did all this sequencing. These sequencing projects were carried out by the Joint Genome Institute, and that is part of the Department of Energy. Something that may have been lost in the whole Human Genome Project is that it wasn't just NIH that got a big bolus of money to spend on these genome sequencing centers. It was also the Department of Energy. And the Department of Energy cares about sequencing because they care about things like mutational damage in response to radiation and stuff like that. So they got quite a lot of sequencers out of this deal, and they've used them to support a lot of interesting research for microbial genomes, for fungal genomes, and also for plants. So it's an excellent resource made available by Department of Energy. I would note, however, that you cannot just anonymously go onto the Phytosome website and download whatever you want. You must create a specific Joint Genome Institute account for yourself. Now this is free, but it may take a couple days for that to be processed. So you should always keep this in mind that if you're going to be even casually browsing in there and expecting to download a data set, you'd better get your account in place first. It's also worth noting that some of the data in Phytosome are not yet freely available for anyone to use with no attribution, for example. So some of the data that you would require that you would acquire from this site will require you to agree to certain agreements before you're able to download a genome that's been newly sequenced, for example. It may be available in Phytosome even before it's published, for that matter. So there are some agreements that are required for specific data sets within there. Now, I wanted to talk about what you're likely to find when you go studying a particular species. So I'm looking here at sorghum. It's a very well-established genome annotation for them. When I go looking for sorghum bicolor, I find S bicolor 454 and S bicolor 730. So these are two different cultivars, basically, that have been independently sequenced and annotated. So very different versions of annotation, but relating to the same species in this case. So you can see which versions these are at, version 3.1.1 and version 5.1. And if you were trying to match your protein identifications from a 3.1.1 sequence database to a genome annotation that resulted from, say, 2.8, you might find that there are some mismatches. So keeping these version numbers square in your mind is very valuable. As with the human genome, the plant proteome, the plant genomes continue to evolve in their annotation over time. So here I'm giving an example of the kind of information you would find in the FASTA file. Yes, it's quite compact. Again, this is something where having the PDF of these slides can help you a lot. But what we see is SOBIC 7G14050.1.P. This is a protein found from this genome annotation. We've got a PAC ID, a transcript number, a locus, an ID, and an annotation or revision. But what you don't find there is a description line. And you don't find the gene ontology codes, and you don't find the interpro hits for this. They do have these, but you must download the annotations as to what these genes represent elsewhere. It's a separate annotation information file. So that information is kept out of the FASTA and kept separately in a different table. If you go looking at this on the website, you'll find all kinds of information like Gene3D, links to putative structures of these things, interpro hits of other sorts, PFAM, COG, gene ontology, etc. This information on the website is not coming from the FASTA, it's coming from this annotation info file. So let's then talk about how we would deal with this in a common context. So imagine that I have analyzed a pair of cohorts, and I now have a list of proteins that I think are differential between my cases and controls, or my well-watered plants and my drought-like watering plants. So for something like that, I probably have a list of accessions for the proteins that are differential, the Q values, these are the false discovery rate corrected p-values, multiple testing corrected p-values, and a log base 2 fold change. So that's really lovely. These are the kind of information I would use for a volcano plot. But assuredly, I would like to know expected functions for any of the genes that have those on this list. But that's not included in my FASTA, so it's not copied to my differences text file. Now I can download the sodium bicolor 454, 311, annotation_info.txt. It's a very long name here. But that's where I'm going to find all the information describing these gene names with gene ontology and so on. That's great. This information is more than I want, though. I'm going to want to grab some subfields out of this. Specifically, I'm going to grab the name of the protein, the gene ontology term, and a description line saying what homolog exists for this in Arabidopsis and how is it described in Arabidopsis. That's one of the ways that you can get this information. Now, I've provided a little bit of our code. I suspect it's not visible on the screen you're looking at right now, but if you download the PDF, you can copy it right out of the file. So here we have basically 4 lines of code that are going to do some magic for us. This is joining together information from our differences list and the annotation info list. So we're going to create a variable called diffs and use the read table function in R to read that differences file into that variable. We're going to create another variable called annot for annotation, and we're going to use the read table function to read the annotation information into that variable. Now we're going to throw away a lot of the information from the annotation file. I wanted to keep just the peptide name, the gene ontology term, and its description line in Arabidopsis. So this subset function right here keeps columns 4, 10, and 13 from the description file. And then finally, I can combine those using the merge function. I specify that I want to merge the data from diffs and annot, our 2 input tables. And in my diffs file, I'm going to keep the accession column. In my peptide info column, I'm going to keep the peptide name. And I'm going to require that these values be equal to each other to be kept in the file. So I run that, and now combo pops out. It has my accessions, my Q values, and my log2 fold changes from the first file. And it contains peptide, the Go term and description lines from Arabidopsis in these last 2 columns. Presto changeo, it's all done. Now you could do this by typing Control F on the database of annotation information and copy those things over individually. But if you have 100 different protein differences, you'll be very glad to have a little script that does this for you. Do you have to do it in R? No. You could do it in Python very happily. You could do it in all sorts of languages. But this is an example of how to do it in a statistical environment, R. Okay, so we talked a little bit then about phytosome and its amazing value, but we should also think about ensemble plants. I'm borrowing some slides here from Alina Mushtaq Stolver over at Ensemble, who had been using these as part of her training slides. She graciously let me use her PowerPoint. So ensemble plants is one area of ensemble, but it's hardly the big one. There's an ensemble metazoa, ensemble bacteria, ensemble protists, ensemble fungi, and ensemble COVID. I think most people are probably familiar with ensemble most for dealing with things like the human genome. Ensemble is a really great database for dealing with these the mammalian proteomes that have been produced and for all kinds of interesting animals. So, but they do not cover just the big furry animals. They also cover things like plants. So ensemble plants is not something that suggests they just got started with this. This is a really consistent effort that's been moving forward for years. What we see is that in release 60 now, they have 240 genomes for 129 species. If you're just asking, do they have more species or fewer than phytosome, I don't think you're really thinking about the value here. Generally speaking, we don't care about the database as having more than another. We care about whether it contains the organism that we care about, right? So it may be that ensemble plants, despite having fewer species than phytosome, may have the one that you care about, and so it doesn't really matter about the other 128 of them. So they have genomes and gene builds just the same as phytosome does. They have genetic variation data, which is awfully useful when you're trying to understand how strain variation alters what you find in the species. Comparative genomics, to look across different genomes, to look at what kind of regulation information we have for how these genes are regulated. It has its own biomart to help you download exactly the data you want to acquire from them. They even have their own tools for that you can download to interact with the data. So it's quite a lot of good stuff. As well as if you're writing software to interact with this, they have their own API for talking to the database as well. So Ensemble Plants has a lot for you. I really felt that this slide was one of the most valuable ones that Alina produced for us. So annotation is a continuing iterative process, happens in multiple releases over time. We refine them and make them better and better. But it's pairing together algorithms and human insight. You should have seen something like this before. For example, if you were working with Uniprot, you probably know that there are Swissprot entries and Tremble entries. And the Swissprot entries have been annotated by humans, have been checked and verified by humans. A lot of software has been used to produce the Tremble annotations, but they haven't yet passed a big check mark by human annotation on top of that. So in the same way, the work from Ensemble is pulling together this information from both algorithms and human insight. So that can come about by using EST libraries, express sequence tag libraries, to confirm that a protein coding gene exists at this site. cDNAs and RNA-seq can be used for stuff like this. They may be using just homology to say, you know, there's a gene between these two in wheat, which makes me think this gene that may or may not be a gene that lies between those same two genes in another species is a real gene by rule of synteny, to say if the gene exists between the two in this species, it may exist between those two in another species. Okay, so homology annotation is very valuable for that. Certainly, having a human in the loop is a very big deal. You could be one of those folks who says that AI is going to remove the need for human curators, but I'm not talking to you. I'm talking to scientists. And for scientists, we know that the insights we provide on the data frequently outstrip what the software can do in the same system. And finally, there's probably some amount of ad initio work going on. So if you're simply looking at a sequence to say, what here looks like it would be a protein coding gene, ab initio algorithms are likely to be providing that information. Now, it's true that Ensemble Plants is focused on plants, but it's certainly not the case that only plant genomes and proteomes have any relevance to agriculture or to plants in the wild. So I thought it's worth talking about some of the ones that come into play here. Triticum aestivum is one that we should know about because wheat is a really, really big deal. Lots of people are eating it every day. So having Triticum aestivum well annotated here is taking place in conjunction with the International Wheat Genome Sequencing Consortium. So pulling together external bodies of esteemed researchers in this particular species makes the information drawn into Ensemble of higher quality. Similarly, Hordeum vulgare is focused on by the International Barley Sequencing Consortium. Again, wheat, barley, very sensible they would show up and have these really great communities feeding information into Ensemble. Ozera sativa is rice, and we're going to talk a little bit about the rice annotation project in a few slides, so we'll come back to that. Now, what about Arabidopsis thaliana? Arabidopsis thaliana is not a species that we eat. So it's, if I remember correctly, a watercress, basically. But it does have a very compact genome, and that's made it quite an interesting species for a long time in plant biotechnology. On that, we're going to talk about the Arabidopsis information resource in a few slides. Now let's get over to the other side of these. These may not be as clear to you. So Fusarium I will be able to pronounce it. Fusarium graminearum is a really interesting species that infects things like wheat crops. So we don't just have wheat involved, we have a pathogen of wheat involved. Zymoseptoria triticii is another pathogen that afflicts wheat. That's what the triticii bit is all about. And that sequence, in this case, has been drawn from the Joint Genome Institute. They do a specialty in that particular species. Remember that I said they do work in fungus? Yeah, good example. So then we have Schizosaccharomyces pombe. Well, I think many people know this as a yeast that we use a lot in research labs, but it also shows up quite frequently in for example, fermentation of species. I seem to recall reading that it had been originally discovered in millet beer. So it's good reason to study these things. If you don't have beer, you know, many people would feel their lives were very much diminished. So I'm glad that we have yeast that can be used in conjunction with plants to produce agricultural products. Okay, and finally, Drosophila melanogaster. Now, Drosophila melanogaster is not a plant, quite obviously, and it's not some fungus, it's an insect. But if you remember, the common name is fruit fly. Well, of course, Drosophila melanogaster has relevance to the folks who do ensemble plants. So you can see that plants and the species that love them can all be brought together under one roof. Okay, if you haven't heard of Elixir, it may suggest you haven't been working in bioinformatics in Europe, because Elixir is a huge infrastructure project that spans many, many laboratories. And one of the things that they've really emphasized is that bioinformatics tools should not be siloed away from each other. These information resources should be tied together for producing a lot more value. So for this, information about structural domains, phenotypes, sequence variants, gene expression, of course, is quite important, biochemical pathways, cross-species interactions. These are all sorts of things that you might expect to be pulled together to make bioinformatics work more coherently. But look at this down here, plastic degrading enzymes. Yes, the plastic DB is out there. There are enzymes that can work with plastics, and so that kind of information gets pulled together under this big hat of Elixir, which means it's all available to Ensemble. If you go back in time to your genetics class, I appreciate these are very, very small fonts. I would just say that we used to have this this sort of shorthand that we would use for genetic variation that would help us to assess whether a sequence change had a big impact or no impact if the mutation took place. Did it affect phenotype? So if it created a stop codon in the middle of a coding sequence, that would be an example of a nonsense mutation. If it changes an amino acid from one letter to a different, that would be a missense mutation. So we have this kind of lightweight ontology of how we classify the impact of different mutations. But the people at Ensemble have taken that a lot further. So what we're looking at here are all the different ways, all the different classifications that they use for describing how phenotype is affected by the change of an individual nucleotide or more. So I think it's really good that they've been nice and systematic about this, because using this information sensibly is best able to proceed when we have good definitions in place. Now, I said I was going to mention Plant Garden. It's a relative newcomer. You can see it was originally popularized in this publication in 2023. So that's only 3 years ago when I taped this. But it's really worth looking at, because a lot of the genomes that appear in Plant Garden are ones that are not accessible in Phytosome or in Ensemble plants. So you can see that there are 680 reference genomes here. So I was not able to get a count on how many different species there are, because I was in a hurry at the time. But the number of species present here can sometimes give you species that you'd love to be in Phytosome or Ensemble plants, but they just aren't yet. So you can download this information, gene sequences, PCR-based DNA markers, trait-linked DNA markers. Obviously, we care about phenotypes. SNPs and insertions and deletions, all that kind of information is available. And they did this all by use of the high annotation engine that they built. And it was a very good strategy to make it possible to combine huge volumes of information about a very large number of plants with strain information to make it available in this way. And the stuff that's available here is all just freely downloadable, no account even required. Okay, so that brings us from databases that focus on a very wide variety of species to talk about information resources tied to two key model organisms. So we start with the Arabidopsis information resource. TARE is a very, very big deal. You can see that this started back in 1999, which might seem like the Stone Ages to some of you, but I remember that as 3 years into the period I was in grad school. So this still feels fresh to me in some ways. So the Arabidopsis thaliana genome was not yet finished when this information hub was created. But this has really helped to nucleate, to pull together a group of researchers very invested in Arabidopsis as a model system for biology. Okay, so I love some of the statements that come out of their work here. So currently, 74% of the proteome has been annotated to at least one gene ontology term. This is magical. I think if you go looking around the different genome annotations that we have for plants, you would be very hard-pressed to find anything that even approaches something where 74% of the protein sequences have been attributed to gene ontology. That's an astonishing accomplishment and really speaks to how many people are invested in making this a brilliant model. In their paper they wrote, TARE's ultimate goal is to maintain a gold standard annotated reference plant genome. I mean, you can't really argue with that. That is a really impressive task that they published back in 2001, and they've gone about as far as you can go and with all the skill sets that we have today. So there's a really key thing that happened, though, in 2013. The funding that had been making TARE go forward all these years dropped away. And they didn't have money to just keep all the people on staff that they'd had in the time leading up to that. So they changed to a subscription model in 2013, which means that if you want to use TARE as an everyday researcher, you're going to have a subscription. You're going to pay for it. And that's daunting for people who are living in under-resourced laboratories. So I want you to keep this in mind when we talk about cuts in science funding. This is the sort of program that we can lose as a result of that. At base here is a really important number, though. I think that's worth talking about from a scientific point of view rather than science policy. Here we have 27,655 protein coding genes. One of the chief reasons why Arabidopsis became such a popular laboratory plant was its relatively compact protein coding gene set. 27,000 is still, let's say, 40% larger than the human genome. We have just over 20,000 protein coding genes in humans. So as plants go, this is tiny. And it's a really nice plant for us to do all kinds of genetic manipulations and so on, and it's not so big that you can't just house it in your lab. So Arabidopsis has been a very popular plant for a very long time. We should also, though, think about the rice annotation project. So this top line, I think, that these two quotes really drive home why rice needs to be prioritized. Rice is considered a model cereal plant, that's something Arabidopsis cannot claim, because of its small genome size, also compact, and a high degree of chromosomal collinearity with other major cereal crops, such as maize, wheat, barley, and sorghum. Remember when I was talking about synteny a few slides back, that we think this gene in the middle is a real one because a gene exists between those two genes in a species we know well? In the same way, rice is similar enough to these major cereal crops that we can count on this degree of collinearity, of sequences being in the same order on similar chromosomes across these species. It's very powerful. And why do we care about plant biotechnology? This bottom quote is a fairly good one to start with. The cereal crops, rice, maize, and wheat, provide half of the food eaten by humankind. Here I believe they're quantifying on calories, basically. So of all the calories humans eat, rice, maize, and wheat are probably producing about half of them. That's a really stunning number to think about. So we obviously do care about cereal grains, and the Rice Annotation Project has a lot to offer here. So they now have 6,371 annotations available for protein coding genes. That's out of 33,000 that are known to code for proteins in the genome. So this is not quite as compact as Arabidopsis. Remember, that was about 27,000. Now we're talking 24,000. Sorry, for 33,000. So we're a little larger than Arabidopsis, but because it's so closely related to cereals, rice has become a model in its own right. Now, I want to talk about a couple other projects that are not the same thing as the Rice Annotation Project. So for those, we have the 3,000 Rice Genomes Project. Again, we would like to know better about the degree of variability in the genome that what genetic variants do we see as we look across different strains. That's something that the 3000 Rice Genomes Project will give us. And you should also not be confused by the fact that not only does the Rice Annotation Project database exist, there's also the Rice Genome Annotation Project, which is a separate project. They have their own website. They're not the same thing. They work together, they talk together, but these are very similar names for different projects. Now, before we start presenting this as if everyone in plant biotechnology is working on a cereal or something that you grow for agriculture, it's important to remember that there are many crops that can be neglected because there's not as much research funding available to deal with them. And for this, there are a lot of traditional crops grown in the continent of Africa that have not really been given a lot of attention by Phytosome or by ensemble plants. So I really appreciate the African Orphan Crops Coalition for pulling together a bunch of different laboratories to produce genomic resources in understudied species. And they've already picked out 101 different traditional African food crops that they want to study. But they've already managed to publish 8 of them, so this is a pretty impressive effort. The apple ring acacia, one of my favorites, the Bambara groundnut, hyacinth beans, marula, horseradish tree, jackfruit, breadfruit, the African eggplant, the shea tree, and the water yam. So these species may not be all that familiar to you. I lived in South Africa for 5 years, and I didn't recognize all of these names. But when I talked to my wife, she said, I fed you about half of those. So this is something that a lot of the foods that we pick up as we move to new parts of the world have a lot of plants involved in them that we may not know about from commercial farming, say in the USA, or in the continent of Europe. So there are lots of crops out there that people eat, that people depend upon, that may not be covered well by these canonical databases. So they've been developing reference genomes for these. They've been producing some transcriptomic data as well, and they're working on trying to gauge variation. Oh, I should mention this picture down here. I couldn't help it. It's my favorite, the Bambara groundnut. So there you have it. So what kind of information are you likely to find as you go pursuing a genome in these resources? Here I'm looking at gigadatabase.org. This is part of, it's basically file hosting for the African Orphan Crops Consortium and a coalition. But we're looking at the data made available for Vigna subterranea, the Bambara groundnut. Of course you are. So I wanted to talk through some of the different files that you might see. Again, the PDF is going to help you read these. At the top we have proteinbusco.zip. Busco is really useful to you, B-U-S-C-O, when you are trying to gauge how complete is the genome for this organism. If you see a Busco score of 74% completeness, that's probably not a very mature genome. You'd love to see a number like 95%, or even better, 99%. And so that information can be found in these Busco output files. You can download them, they're just text, and you can then look through the report to see what kind of completeness results. You will sometimes see files with a .fa extension. In this case, it's a transcript.fa file, which is to say this is a nucleotide sequence database in FASTA format. So .fa will frequently be shown for FASTAs, whether they're nucleotide or protein. Now, if you were looking at a resource at NCBI instead, in the United States, then you'd probably see something that says FNA, so a FASTA of nucleotides, whereas a PEP for them, a PEP, is a FASTA in protein sequence. So you may see lots of files that are all FASTAs, but they may have different extensions to reflect where they came from. You'll see some GFF files here. These are general feature format. They're very useful for understanding which aspects of this genome sequence have been annotated to any sort of domain, even outside of the protein coding domains. So a GFF is likely to give you that info. Down here we have a .pep file. Here they're using .pep as a FASTA format in amino acids. So they use .fa for nucleotides here, and .pep, like you see, well, FAA is another one that you sometimes see for protein sequences in FASTA format. And down here we have a CDS. These are coding sequences. So CDS sequences are often given in DNA basis rather than RNA, and they provide us what the coding sequences are for protein coding genes. So this type of information is quite typical of what you see, whether you're going to phytosome or ensemble plants or whatever. You're likely to see this class of files made available as part of each genome. Of course, it's very valuable to you if you can benefit from data other people have already collected in a species you care about. OmicsDI is a very good friend for you. OmicsDI is a website you can reach. It's just omicsdi.org down here. And they've done a lot of work to make searching for data sets in biotechnology a lot easier. So I asked the question, how many data sets have been posted in proteomes for alfalfa? I chose alfalfa mostly because it's a lot of fun to say, but that's fine. Alfalfa is a great species for this choice. If I do the search for alfalfa, I can see over here that I have many different technologies that have been used in the species. Genomics, transcriptomics, proteomics, metabolomics, and some that have just been annotated as other. If I click on proteomics, I get back 19 different listings for studies that have done proteomes for alfalfa. And if I click through here, I can generally get to the raw data that correspond to that study, as well as stuff like what paper was published in connection with it. So OmicsDI can make it a whole lot easier for you to reach immediately to the set of data sets that are already existing with raw data supporting them, whether it's coming from sequencers or coming from mass specs. There is a penchant in biotechnology for people to assume, at least in the world of proteomes, that UniProt knowledge base has everything you need. But I gotta tell you that if you look for all of the species we've discussed today, some of them, yes, you'll find good reference proteomes in place, but some of them you won't. And the reason for this is that UniProt is trying to serve every research community. This is a difficult task, and plant biotechnology sometimes has lagged behind others in the support that it gets in the omnibus web resources out there. But there are quite a few plant-specific web resources that can help you that are specializing in your species or in a group of species relating to yours. So it's definitely worth your while to step beyond NCBI, to step beyond UniProt, to look at plant-specific web resources if you want better coverage. It's clear that phytosome and ensemble plants are have tremendous bioinformatic resources to bring to bear, and they're funded quite well as well. But if you're working in a species that is of relatively rare study, like the Bambara groundnut, you're probably going to need to look elsewhere to find a proper genome annotation. Do not assume that it doesn't exist just because it's not in ensemble plants or phytosome. They try to be as inclusive as quickly as they can, but a lot of species have not yet made it in that have genome sequences available. You should not fall into the trap of thinking Arabidopsis thaliana is the only model plant genome. Certainly if you're working in cereals, you should probably be thinking about rice as your model as well. And I would point out that the rice annotation is not a new kid on the block. They've been working on this now for 20 years, so I think it's definitely worth your while to look at the solid annotation information available for that species. So new annotations crop up every day. If you're working on a PhD, then you're probably working on this project for at least 3 years. Over the space of that time, if a genome annotation isn't already available, you may discover that it has become so in the meanwhile. So you should always keep in mind that when you see new data sets cropping up in the sequence read archive at NCBI, for example, for your species, that's a pretty good indicator someone's working on the genome. Maybe you should reach out and touch them and say, hey, is it okay if I take a look for information on this particular gene that I'm really interested in? You could be part of that annotation project. So new stuff is happening all the time. It's very good to keep focused on that. I've provided my email address here. If you need to reach me, please do send an email. It is okay to send me another email a week later saying, did you get my first one? Because I get a lot of email and I don't always stay on top of it. So please bear with me and I'll try to get back to you, okay? Well, so I hope that this has been a useful tour for you of all the fine information that you can find in web resources for plants. And I hopefully will be back with you again soon to talk about what you can do when there isn't a genome to download for your species. So you can look forward to that video in the near future. All right, thank you very much.